necessarily lead to an enhancement of therapeutic efficacy, and it must be specifically claimed and established by research data. And so when we talk about efficacy and evidence, which stage? Because when we do in the pharma, there are different, you have basic research, you have preclinical research, and then it is IND, then you have phase 1, phase 2, phase 3, and so you can have many types of data, PK, PD studies, PK in humans, sometimes PD, you have PD studies. Which data are they looking for? And that is why which data will be sufficient to show enhanced efficacy. I just also want to touch on section 3E and 3I because they're very relevant for the pharma industry. We've had a recent guidelines that has been brought out by the Indian Patent Office. We've had 2 rounds of discussions, and we're quite happy that the Patent Office is discussing with the stakeholders, though a lot of the things that we did put up were not accepted by the Patent Office. One of the things we are very happy about is, even if you have a patent application and the claims for a novel compound, they don't give you a composition claim. And they would say it's a mere admixture. So we really, all the stakeholders did come together and we said, no, if the compound is a novel compound, the composition should also be allowed because the composition is novel. So that way we are quite happy because the Patent Office has come in October 2014 and said, if you put 2 compounds which are old integers, so we are very happy because this gives guidance to some of the controllers who would now accept this. If you look at the pre-grant opposition that came out yesterday on the Gilead cases on Sovaldi, these people during the hearing have cancelled the claims on a composition even though the compounds are novel, because the controller would have said 3E, 3E, 3E. So this guidance that we get, the guidelines which have come out from the Patent Office, makes it very clear that if your compound is a novel compound, you can definitely claim for a composition. Earlier on, they would also want us to give the percentage of all the ingredients, and we said it is too early in the research to actually tell you how much is the percentage that you need of all the active ingredient and the excipients. So at least that guidance has come that if it is an old integer, you need to show that it is not a mere admixture and you have some synergistic properties. But if it is a novel compound, you can get a composition claim. Now this is very good because what happens if during the prosecution you do cancel your composition claims and infringer does make a composition claims, they can always come to the court and say, look, you had composition claims, your composition claims are out, and therefore we are making the composition, because you must realize the judiciary is quite new to this cases in pharma, and they could get swayed. Therefore, we are very happy, at least with this, that there is some guidance, and the guidelines tells us that with new molecules, you can get compositions. Therefore, compositions with novel compound will not fall within the purview of Section 3E. The other problem that we have is assays, diagnostic assays. Even if you do a PCR assay or you do a ELISA assay, the Patent Office is not allowing under 3I. Even though our Section 3I says it is for a process of treatment, any process for medicinal treatment, surgical treatment, curative treatment, diagnostic or therapeutic treatment, so it should be read with other treatments. Just like they did in 3D. 3D clearly says that it has to be a derivative. And so here they are not reading, they're reading every word. Unless we get good case laws, we will not be able to actually challenge this. So right now, any diagnostic assays, any method, any genetic marker assay, any assay that we do, even though it is not carried out on the human body, is actually being rejected by the Patent Office. And the guidelines does nothing good to us. It does not tell you anything better. And so we have to keep looking around for 3I. This is a challenge. Though in the EP you do have on the human body, take any assay carried out on the human body is not, but we don't have it in India, and we still have this problem. I just wanted to bring up this case which has come up yesterday, rejected by the Patent Office on a pre-grant opposition of Gilead. This is the Sovaldi case, and here the molecule is novel. It has got over the inventor step. It has been granted in at least 15 countries, And look at the state in India, it has been rejected, and rejected under 3D without much explanation, to say that this molecule was known and this molecule is a derivative of that molecule. It's just like that. Under section 3D, you have to show that it is what is a known substance. You have to show that it is a new form of that substance, and that new form, in order to be patentable, has to demonstrate therapeutic efficacy, and therapeutic efficacy has to be established by means of filing comparative data. And now the applicants did argue that these are not new forms of known compounds, but the controller has rejected this application. And this is what they said: it is unexpected that appending both a methyl substituent and a fluoro substituent to the 2' position of a nucleoside, wherein the methyl substituent is in the beta position up and the fluoro position in the down position, produce a compound 5, which is of this application, and it is both a potent inhibitor of HCV, hepatitis C, and a replication in cell culture, and lacks cytotoxicity. Whereas the similar compounds, compound 3, which is the 2 beta position, a fluorine in the 2 beta position, or in the 2 alpha position, you have compound 2, and you have a difluoro in the 2 position of compound 1. They demonstrate activity against HCV, but they also show significant cytotoxicity in 1 or more cell lines. Even after giving this explanation, and it is not, it is a novel compound, we are facing problems. I'm sure it's going to be appealed, and once we get this, because even in the Tarceva case, Roche was a Cipla. They have made it very clear that even though there's a minor difference, it is a new molecule because it cannot be a derivative, because by making these changes, it was not able to get into the active site, and therefore it cannot fall under 3D. Even though Justice Prabhadevan in the IPAB has made it very clear, the Tarceva case is also very clear, but we still have got rejections for this Gilead case. So what are the things you have to be very careful about? There will be some amount of uncertainty of what is this increased efficacy, how do you prove it? Now, when you do have a very broad Marcus structure, you have to be very careful. They're not going to allow that. They want your Marcus structure to be restricted to what, to the disclosure, to the different molecules or compounds that you have disclosed, very much like the practice in the Japanese Patent Office. Any broadly claimed Marcus in prior art may severely affect patenting in India, because there's a little problem here versus disclosure versus enablement. Even if you have a Marcus, they say, oh, that's already there, even if you didn't disclose it or enable it. So this is one case law that we really need about what is novelty destroying. All over the world, it has to be disclosed and enabled. In India, it doesn't seem to be, and that is a serious problem that we have. If patenting in India is considered important, even Indian entities may have to redesign their R&D, and they have to test for enhanced efficacy will become necessary. Now, the other thing is you need to submit data even after you file, whether it's animal studies, and look at the efficacy data. And this is all I have as far as patenting in pharma is concerned. Thank you very much.